Our guest speaker today is Gina LaLiberte from the Wisconsin Department of Natural Resources. Gina is passionate about algae, and she is our state blue-green algae coordinator. Her presentation today is titled Algae and Other Little Green Things Are Fun. Gina, I will stop sharing my screen and I'll turn it over to you.
Thank you very much for inviting me to talk today. It's really cold and snowy out, and hopefully seeing a lot of these little green things is going to put you in the mood for spring. Our days are lengthening, thank goodness, and it will get warmer. Just doesn't feel like it right now. So anyway, I'm going to talk about algae and other little green things that I think are a lot of fun, and hopefully you will too. And I'm going to try to target a number of species and organisms that are common in the state and that you're likely to encounter as either a citizen lake monitoring network volunteer or a water action volunteer. All right, there. And so you can see my next slide. So one of the things that's really going to help you in appreciating a lot of these little green things is being able to magnify them. So do that either by getting yourself something like this hand lens or a jeweler's loupe that you see on the left, or there are also a lot of inexpensive field microscopes such as this one on the right that are about $20 to $30. There are also some really advanced ones that connect to cell phones for imaging and that can do image capturing as well. So magnifying is really going to help you to appreciate what you're seeing. Another thing that's really going to be useful for you is something like an underwater viewing bucket, such as you see being used by Matt in this picture. You can either make your own or you can use a commercially available version. And I just want to make sure, yeah, you can still hear me and everything correctly.
Yep,
correct?
You sound good.
Okay, great. All right, so underwater viewing buckets. This is one that we made at the DNR for some of our work activities, just using a commercially available storage bin. We cut a hole in the bottom and screwed and caulked a piece of plexiglass into the bottom of that, and that's really great for seeing what's growing on the bottom of aquatic habitats. For example, this photograph of Vaucheria, which I'll talk about in a little bit, that was made just by putting a point and shoot camera right up against that plexiglass and shooting straight through, and it looks like a fancy underwater camera basically. So that's a do-it-yourself version. There are also commercially available versions that either look like a 5-gallon bucket with the plexiglass in the bottom, or sort of a traffic cone style where you all have almost like a scuba type mask that you look into, and then towards a wider lens at the bottom. And those commercially available versions are about $30 to $40. So that's a really great way to see the things in the waters that you're monitoring and really start to appreciate them. So once you start seeing those things on the bottom, how do you actually identify what you're seeing? So I've got this website. This is the University of New Hampshire's Center for Freshwater Biology. They have a number of photographic keys online, including some basic cyanobacteria, aquatic plants, although I highly recommend, if you're working in Wisconsin, to either get Through the Looking Glass or Paul Skowinski's excellent photographic book of aquatic plants of Wisconsin. Those are available from UW Extension. This Center for Freshwater Biology also has great keys for phytoplankton, vernal pool plankton, stream insects, and zooplankton if you really want to get into identifying some of these organisms in aquatic systems. And there are also a lot of books and other resources that you can either purchase or available free online. Some of these titles that I have pictured here are free at Biodiversity Heritage Libraries. That's biodiversitylibrary.org. I do have a link of a number of resources, including the keys from University of New Hampshire, some downloadable books from biodiversity library, and a number of other algae identification resources that are online. And Paul is going to provide that handout to all registered webinar participants after the meeting, I think, when he sends out to the link to the webinar. Now, if you are watching the archive version of the webinar and you aren't able to get that, that handout, please contact me with my email that will be repeated again at the end of the webinar, and I can get that to you. So those are some of the resources that are going to be really useful for you for identifying algae and a lot of other little things in lakes. And if you haven't checked out Biodiversity Heritage Library, do so. It has all sorts of great antique and relatively modern natural history books that are free and available online as PDFs. So it's a really cool resource. So as the state harmful algal bloom coordinator for Wisconsin, most of what I deal with is blue-green algae or cyanobacteria. So I am going to start a little bit with cyanobacteria, but that's not really going to be the focus of my talk because unlike me, most people don't think that they're very fun. There are a number of cyanobacteria identification resources online. In particular, I have both a recorded workshop and last year's recorded presentation at the Wisconsin Lakes and Rivers Convention. Those are at the convention archives, and I believe Paul is going to share links to those either in the chat or via the follow-up email after this webinar. So I'm not really going to go into great depths about cyanobacteria, but I am going to start with them because they're the first photosynthetic organisms on Earth about 3 billion years ago, and you can really thank them in part for having oxygen in our atmosphere. And they're maybe not fun for most people, but their ecology can be really complicated and really interesting. One important thing to keep in mind is that cyanobacteria or blue-green algae are in all water bodies. They grow even in those that have really excellent water quality and low nutrients. They're just different species that tend to be there instead of the species that you normally think as the bloom-forming cyanobacteria or blue-green algae. Now, when planktonic cyanobacteria, such as that planktonic means basically floating in the water, such as those that you see here are actively growing, they tend to be more mint green in color. So they look like these little tiny green specks that you see in this water. Sometimes they look like a pale green dust on the surface, or there are some species that can even look like tiny grass clippings. So just sort of a bird's eye view of what planktonic cyanobacteria look like on the surface of a lake. Now, if you have really green lake water, for example, how can you tell if that's planktonic cyanobacteria or something else? You can do what's called the jar test to see if it forms a floating layer. Now, this test and the stick test, which I'm going to talk about next, they aren't completely foolproof. The only way to really know for sure is to put the green stuff under a microscope and look at it and confirm that it is or is not cyanobacteria. But this is a good way to kind of get an idea of if you see a lot of green stuff floating on the surface of a lake, making that water green, is it planktonic cyanobacteria or not? So basically what you do is you put the green water in a jar, cap it, shake it up, and just wait. Planktonic cyanobacteria float, and so eventually they will float to the surface and form sort of this floating layer of green stuff on the surface of the water in that jar. True algae that aren't planktonic cyanobacteria tend to sink to the bottom, as you see here and over here. And there's more information at the Department of Health Services website on their blue-green algae page. They have a number of resources on the resources tab. And I think that link is also going to be provided to you after this webinar. So this is what the jar test looks like. So You do have to look closely at those floating green layers because some of them might be plants. So again, here's that photo from the previous slide. You've got that floating green layer of Planktonic Cyanobacteria. Here you've got another floating green layer in this jar. So what exactly is it? You do need to look closely because there are tiny aquatic plants that can form a floating layer as well. So here we are looking through the side of that jar close up. You've got a lot of little tiny watermeal plants. Here you have a duckweed plant with roots, and here we are looking at it from the top. Duckweeds and watermeals are tiny floating plants, and the CLMN webinar series from last year, Paul Skowinski did a webinar on unique plants in and around Wisconsin lakes. So if you want more detail on duckweeds, check out his archived webinar from February 24th last year. So here you see a lot of little green plants floating. These larger ones, relatively speaking, are duckweeds, and duckweeds again are going to have a root that hangs down that you'll be able to see from the side of the jar. Watermeal plants don't really have roots. They're just little tiny ovoid firm plants, and they feel kind of gritty if you rub them in between your fingers. They have a very regular outline, as you can see here close up. They're about the size of a third of a sesame seed, so really, really tiny. So again, you can do that jar test, but familiarize yourself with what duckweeds and watermeal look like, and do look closely. Just because something is floating on the surface of the jar doesn't mean it's necessarily Planktonic Cyanobacteria. Now, something else that you might mistake for a floating layer of cyanobacteria is pollen. Pollen, especially in lakes, can really look like a cyanobacteria bloom on the surface, but pollen tends to be pale yellow, as you see here in this inland lake, here forming sort of a yellowish scum and floating on the surface. And here you see a whole lot of pollen washing up on the shore of Lake Superior. So one of the key things to do if you think it might be pollen is look for similar yellow dust on land. Especially car windshields, they're going to get really yellow and dusty. You can write "I love algae" in that pollen dust on the surface of the car, and that will kind of help you to confirm that, okay, I'm seeing this yellow stuff in the lake, but I see a lot of dust on land too, so that's probably pollen. So cyanobacteria can also form floating mats in water. So how do you tell if those floating mats are cyanobacteria or something else? You can do what's called a stick test. Grab a stick, put it in the water and pull it out. If that stick ends up coated with something that looks like paint, like you see here on the lower left, that's probably going to be cyanobacteria. However, if that mat material ends up draping over that stick like green hair, like you see here in the middle, or in this really huge example on the right, that is going to be filamentous green algae. So that's a stick test. I will caution you, however, there is one exception, so you do have to look closely at the color. There is a cyanobacterium called Microcyra that you see right here. It's formerly known as Lingabia wolii, and that looks, it's a really super dark green, almost looks black with the naked eye. And these are huge filaments for cyanobacteria, so up to several centimeters long. So hypothetically, they could drape over a stick, but you see this big color difference. This looks really dark, almost black. Filamentous green algae tends to have this really nice green color. So not only look at whether it drapes over a stick, but look at that color as well to see if something that drapes is cyanobacteria or filamentous green algae. All right, so that's it for cyanobacterial blooms for now. So I'm going to move on to other green stuff that, I mean, this might not look terribly impressive to you, but there's actually some really cool things in there that you can see if you look closer. And that is the Carophytes, so species in the genera Spirogyron and Zignema. So these are filamentous algae. They secrete pectin that gives filaments sort of a slippery texture, really not unlike say conditioner in hair when you're washing your hair. And that pectin that gives them that slippery texture is one of the reasons it has the common name of water silk. Down at the bottom, we've got the typical life cycle of Spirogyron and Zignema. Typically, they start out growing near the bottom of lakes and quiet areas of rivers in the early spring when the water's really clear and really cold and there are enough nutrients for it to grow. So it kind of looks like green clouds on the bottom. Now they're getting a lot of sunlight on the bottom because they're getting the sun's penetrating that clear water. They're photosynthesizing a lot, putting out a lot of oxygen that may bubble up in between their filaments, and that will lift them to the surface. So you kind of get this sort of green bubbly appearance of mat, and that's what gives the Spirogyron Zignema their other common name, frog spit. So kind of this bubbly green stuff on the surface. Now, when it's growing here up at the surface, they're getting a lot of UV light that tends to stress them out. And eventually it gets to be too much for them. Maybe it's warmer than they like up at the surface. There's too much sun. So what they do is they go through conjugation. They go through a process of sexual reproduction. They form what are called Zygospores, basically these resting cells that have this brownish color, and those mats of filamentous green algae up at the top starts to break down. So it kind of looks like this at this stage as these mats are decomposing. That can be really stinky and kind of a nuisance, but eventually these mats do dissipate and break down. But as they're sort of more in these early stages, they're really cool to look at, especially if you've got a light microscope and you can really appreciate the structures that give them their names. In the case of Spirogyra has these beautiful spiral chloroplasts within the cells. Zignema is really cool because it has pairs of star-shaped or stellate chloroplasts within their cells. So these are really interesting things to look at with a light microscope if you have access to one. So that's Spirogyra and Zignema, both of which are very common in Wisconsin, especially spring into early summer. So here is another alga in a different phylum called Ochrophyta, and this is Vaucheria. This is interesting because it has a green color and looks very much like other filamentous green algae, but unlike Spirogyra and Zignema that I previously talked about, this doesn't have any cross walls. Those filaments are what's called cenocytic. They're basically like a big tube. Sometimes there are constrictions, occasionally at some branches in these filaments, but they're basically big tubes, mostly unbranched or sometimes really sparsely branched. And these are also really interesting to look at with the microscope because all of these little discoid platelets in here will slowly move in this process that's called cytoplasmic streaming. And I've got a YouTube address down here where you can also do a web search for cytoplasmic streaming in Vaucheria and see what that looks like. I see Vaucheria typically in pretty clean water streams. They tends to be really cold and very swift streams where it grows, or sometimes on damp soil because they do have a need for pretty high oxygen. But one of the interesting things about this, one of its common names is waterfelt. But if you pet them, basically, these filament clumps feel like wet cat fur. So it's if you're out, especially for wave volunteers, if you're out at your stream, it's totally reasonable to grab a handful of green stuff and pet it to see if it feels like wet cat fur. So that's one of the key field characteristics for Vaucheria. So here is a branched filament. This is a green alga called Clydophora. So Clydophora is really common both in streams and lakes in Wisconsin, especially in the Great Lakes as well. You need a microscope really to confirm its identity, but usually if you are looking at highly branched green alga that kind of feels like wet cotton that you found growing on a hard substrate, that's usually going to be Clydophora. There are some species that can be really large and coarse and have few branches, but this is Clydophora glomerata, so pretty common in streams and especially in the splash zones growing on rocks and riprap in lakes. So here are some new filaments. You can see how bright green and highly branched they are. Here are some Clydophora clumps growing on the bottom of a stream as viewed with one of those viewing buckets. And you can see that this looks really brown, not at all like this nice green color on the left. And that's because Clydophora has a pretty coarse cellulose cell wall, and that makes a really good substrate for other kinds of algae, such as diatoms. Gives them a surface to grow on. So here you see a close-up of some of those branches of Clydophora, those filaments. You see all of these little brown dots on there. Those are individual diatom cells. So diatoms are another kind of algae. They're growing on other algae, so we call them algal epiphytes. It's basically very similar to, say, orchids and other epiphytic plants growing on a tree in a rainforest that just happens at a really small scale on algae with other algae and other organisms. So sometimes you'll get diatoms that stick directly onto the filaments. And sometimes you see down here at the bottom, you have diatoms that grow on these long mucilage stalks to kind of get up into the water. They're not trying to keep from being shaded by others. So these can be really complex and interesting tiny little ecosystems in there. And Clydophora quite often is going to look sort of golden brown to even dark brown, just because it can have a whole lot of other algal epiphytes growing on it. So here is a green alga called Hydrodyctium reticulatum, and this is something that you can identify with the naked eye. And you can see why it has this very distinctive net-like appearance, and that gives its common name, water net. So these are interesting because each of these cells in this particular, so each of these sides of the hexagon in that net pattern, each one of those, as that colony grows and then starts to fall apart, each one of those can grow into another colony. So sometimes you can see really high concentrations of Hydrodyctium or water net in nutrient-enriched systems. And here you see a smaller colony next to the point of a pencil here. This is one close up, and these are Hydrodyctium colonies that were growing in a stream receiving effluent from a wastewater treatment plant. So here's one of those tiny initial colonies, and this is a great big one. So I think this is about 2 to 2.5 feet long. These can get to be really big if there are enough nutrients to fuel their growth. So this is water net. Kind of cool to see it, and you can tell what it is with the naked eye. You don't even necessarily need a hand lens to tell what that is, just because of that really distinctive net-like appearance. It typically grows in higher pH systems. Now here is another green alga, but this is one that typically grows as an epiphyte on vascular plants. This is called Chitophara. It has a couple of different growth forms, one of which is these really small, firm, gelatinous lumps. They can be really difficult to compress. So if you, say, pull one off of a plant, as you see here growing on a piece of decomposing cattail, and you try to smash it under a microscope slide, quite often you'll end up breaking your cover slip just because these are so firm. This is what one looks like that I did successfully smash. You can see that within that mess of filaments that are all semi-connected at a sort of central point, you do have individual filaments with a lot of branching out at the tip. Sometimes, so in addition to these sort of globular forms, Chitophara can take this highly branched or dendritic growth form as well. And here's what it looks like close up. So these are pretty cool. You can tell what they are with the naked eye, but you're only going to see this really fine branching within these accumulations if you use a microscope. And they typically grow on rushes in lakes or cattails, but they can grow on other plants as well. So here you see more of that globular form of Chitophara growing on some mosses. And here you see Chitophara growing on water lobelia or Lobelia dortmana. And this is one that's got that really cool branched form. So that's Chitophara. So we do have some red algae that grow in fresh water, and a number of species grow in the Great Lakes region. Most reds typically are marine, so we would know them more often as seaweed, but we do have some in the Great Lakes region, and this is one that is macroscopic and grows in Wisconsin. This is called Betraychospermum, which translates as frog eggs, and that's because the filaments sort of have this almost beady-like texture. It can be kind of slippery and rubbery. It's kind of an interesting alga to touch. And superficially, Betraychospermum can resemble moss clumps. So here you have 3 clumps of Betraychospermum in this stream. So these are several centimeters long. Here's a clump of moss, and here's another clump of moss called Fontanalis. I'll talk about that in a bit down at the lower left. So typically Betraychospermum tends to be sort of olive green to brownish in color, and has this beady appearance that you can see with the naked eye, or you can see that really well if you're looking at it with a hand lens as well. And that sort of beady appearance is because it grows with branches sort of whorled around a central axis. So here's the central axis of that Betraychospermum alga, and each one of these little whorls is kind of this beady appearance part on this more macroscopic view. And this is what Betraychospermum looks like really close up under oil immersion with a good light microscope. So you can see those individual cells, and sort of a brownish color and a little bit more of a hint of maroon. So you can see more when you get in magnified really closely, more of what you would think is a color associated with a red alga. But this is Betraychospermum. I typically see this in pretty cold, clear rivers such as trout streams, but I've also seen it in the upper parts of the Fox River early in the spring when the water's still pretty cool. So Betraychospermum, a red alga. So I'm going to cover a few more cyanobacteria. These aren't necessarily things that form those planktonic blooms like you would think of. There are some cyanobacteria that can form benthic, which means growing on the bottom, or floating macroscopic colonies. And this is one of my favorites. This is called Phanatathystagnina. So typically it would grow on the bottom of clear lakes. Here's a great photo of a lake in southern Wisconsin from Paul Skowinski. And these colonies can be pretty big for cyanobacteria. I mean, they're macroscopic. They can be up to a few centimeters in diameter. They do superficially look like some other species of cyanobacteria, so it does help to have a microscope in order to identify them for sure. What you're looking for are these small spherical or ovoid cells. Tends to be a little bit more ovoid in the Phanatathystagnina, and a nice bluish green color to them. And the colonies themselves, you can see they can either have that bright bluish green color or be a little bit more olive green as well. And these, they are growing in sort of a gelatinous matrix, and it can be firm, but it's not so firm that you can't pinch through it with your fingers. So that's a Phanatathystagnina. And here's another cyanobacterium. This is called Nostoc communi, and it is actually terrestrial. We can typically see it in sort of wet areas along streams, or even in more upland areas, typically in places with sort of acidic sandy soils, or I frequently see it in gravel parking lots as well. And this is really interesting because it tolerates being dehydrated really well. So here on the lower left, you see what it looks like when it's all dried out. It looks really black and crispy. Doesn't really look like anything except for maybe some broken up dried leaves, or maybe even goose poop if it's lurking in between some grasses near a lake. So when it's rehydrated, when it's full of water, which you can be after a rain or after a really dewy morning, you can see what that looks like here, just this weird rubbery looking green stuff. And that has inspired some of the common names for it. In the past, Nostoc communis has been called star jelly or witch's butter, just because no one thinks about looking at something like this on the ground. It's just whatever, crunched up leaves. And then this stuff seems to appear overnight. So obviously it must be some sort of weird jelly that fell from the stars. I like the French common name for it, crachette de lune, which translates as moon spit. So the moon spat this out and landed on the earth after a night, and just sort of seems to appear overnight. But that's really interesting. And they're really cool if you look at them, look at a piece of Nostoc under a light microscope. So within this green colony and this really super firm or rubbery mucilage, what you see is this kind of yellowy stuff here, that contains lots and lots of these filaments that look like little strings of beads. And I don't have any pictured here, but Nostoc communi is a nitrogen fixer. So like other nitrogen fixing cyanobacteria, it's able to take atmospheric nitrogen and convert it to a form that's more readily used by cyanobacteria and algae and plants. And Nostoc communi is one of my favorites. I always look for it in gravelly places. And this is actually a photograph from Christmas a few years ago at my parents, some Nostoc communi growing next to their driveway. So not terribly festive, I guess, unless you're me, but it's one of my favorites. And I always like seeing it, whether it's the dried up crispy stuff or the fully hydrated star jelly. So I'm going to talk now about a couple, just this organism that isn't necessarily an alga. It's a protist, and it's a colonial protozoan that actually has a symbiotic alga called zoochlorella that's contained within the cells. It gives the colonies their green color. So these Ephridium colonies tend to have a really soft gelatinous texture. Typically find them in more alkaline lakes. So here you see some of these Ephridium colonies attached to Cara or muskgrass plants. Sometimes they just grow sitting on the lake bottom, or sometimes they can break free and float. They're pretty soft, you can pinch through them. And again, like a lot of these other green blobs that you might find in lakes, it's really useful to look at them with a microscope. And then you see that these are what some of the individual Ephridium cells look like. So these protozoans, this little organelles with them, they'll be kind of greenish colored, and they can sort of expand and contract. So that's Ephridium. So now I'm going to talk about an alga to look out for. It isn't necessarily green in color, but it is something to be on the watch for, especially if you're one of the water action volunteers in northern Wisconsin. So this is a diatom. It's a kind of algae. Its scientific name is Didimosphenia Geminata, or we also refer to it as Didimo, and its common name is rocksnot. This is actually an NR40 prohibited species in Wisconsin. So it really helps DNR if it's collected for verification. And I'm going to give you some hints on what to look for in a couple of slides. There are other diatoms. So the reason it's called rocksnot is Didimo is a diatom species that puts out a lot of stock material, so much that it forms these big mats that you see as this orange stuff on a boulder in Montana. But there are other diatom species that are related to Didimo that can also put out a lot of mat material. So you really have to look at it with a microscope or a really good hand lens in order to tell if it's Didimo or not. So for example, here are some fluffy stalk material of Cimbella, another kind of diatom, on a dock piling in Montana. Here are some diatoms growing as sort of an orange scum on this boulder in Lake Superior in Michigan. These are all, of course, my vacation photos. This is what I do on vacations. I take pictures of gooey algae stuff. But this is Didimo in Montana. Unfortunately, it is all over the place around Glacier National Park, and that is in part because the rivers around there are really great for fishing. And unfortunately, anglers using felt-soled waders have really helped to spread Didimo around the park and within the park itself. So if you have felt-soled waders, I would strongly urge you to find something else to maintain your traction if you're fishing or wading, because that felt is a really good way for aquatic invasive species to get embedded and the felt doesn't dry out, and then you can spread them to other aquatic habitats. So strongly urge you to get rid of felt-soled waders and use something different. So where I think you're most likely to see Didimosphenia is in Lake Superior or Lake Superior tributaries. And that's because while it has been found to be growing on the lake itself, it's been pretty well behaved for about 10 to 15 years. However, in 2018, there was a population that has seemed to form nuisance growths in the Poplar River at Lutsen in Minnesota on the North Shore. So that was 2018. And fall of 2021, researchers from the Minnesota DNR, the Science Museum of Minnesota, and UW Oshkosh did a big survey of North Shore tributaries in the lake shore itself, mainly along the Minnesota shore, looking for populations of Didimosphenia. So they are working on trying to figure out why it seems to be expanding into tributaries along the North Shore. It would definitely help to know if it's in Wisconsin's tributaries as well. So if you are a wave action volunteer, this is something to look for, and I'll give you some hints in a minute on what to look for. And so this is what Didimosphenia looks like under the light microscope. This is a Didimosphenia cell. It's absolutely huge for a diatom. Back when I was a phycology student, this was sort of a big game prize. We went to Lake Superior to sample to see if we could find it, because it was so rare at the time, and like a cool big thing to see. Although now, unfortunately, something seems to be causing it to grow to nuisance levels. So we don't know yet if it's some sort of mutation or if it's some sort of really subtle response to climate change. So this is what you're looking for. They are huge cells, and you can actually see them with the hand lens. You won't see them in this detail because this photograph is taken with a light microscope. But what you look for is this really strongly set off head that has this narrowed neck and a really strongly set off foot with sort of this narrowed ankle on the cell. The other species of diatoms that are most likely to form a lot of mat material or stalks are Cymbella, which is sort of look like a half moon shape, or Gomphoneus, which is going to be more club shaped. So here is another Gomphoneus cell under the light microscope. You see it does not have that really constricted neck or that really constricted ankle. So that's what you look for with a hand lens or a microscope to know if you have Didimosphenia or a different diatom. So just be on the lookout. But if you see something that looks like this sort of brownish or orangish scum on a rock in Lake Superior, don't panic, because it might actually be something like these native Gomphoneus species or Cymbella, or you might have other associated things like this Henia superiorensis in there. So there are other diatoms that could be in there as well. So now I'm going to finish up with some small plants that are probably underappreciated in aquatic systems, but are still really cool. These are Bryophytes, so mosses and liverworts. So mosses and leafy liverworts, you're looking for little tiny leaves on the plants, and Phallus liverworts, you're looking for kind of ribbon-like bodies. Especially in mosses, you can see a lot of algae and detritus accumulating in the submerged plants. So you might not see a lot of green plant material. It might be a lot darker just from debris or epiphytic algae growing in there, and you might only see little green growing tips on those plants. So I'm gonna finish up with 4 examples of the easiest aquatic or semi-aquatic bryophytes that you can identify macroscopically or with a hand lens. So I'm going to start with this moss called Hygroamble Stygium 10X. The plants tend to be really large, coarse, they're pretty rigid and dark colored. And if you look at the leaves with hand lens, you'll see this really strongly set apart midrib. And this is a really important field characteristics because you will often see those leaf midribs persisting on older portions of the stems. The rest of the leaves have decomposed. This is an older portion of older growth of that stem, but you still see those leaf midribs. And here you can see kind of those real spiky looking things are leaf midribs on this dry herbarium specimen as well. But here you see those kind of the green leaves out towards the tip. And again, this is that photograph of that example of Chitophara, the green alga, growing as an epiphyte on these Hygroamble Stygium 10X plants. This is another moss called Fontinalis. This Fontinalis tends to have larger leaves than other aquatic mosses, and there are a few species that you can find in Wisconsin. These clumps can be several centimeters long and sort of stream in the current. So here is a close-up of what some of the leaves look like, and then you can see little branches coming off the plants. And again, like a lot of these other aquatic bryophytes, they'll tend to be sort of brownish just from the buildup to debris or other epiphytic algae on those plants. And sometimes you'll only see the green tips out towards the new growth. So this is Fontinalis, an aquatic moss. And here is one more aquatic or semi-aquatic moss called Fissidines. There are a number of species that grow in Wisconsin. Fissidines Grandifronds you typically will find in submerged area and streams, and these tend to be really large, robust plants. Tends to be in more areas with more calcareous bedrock and can be encrusted with marl. But Fissidines plants are sort of flattened like a fern. So you can see that really well in these Fissidines plants just from the margin of a stream. So the plants are sort of flattened like a fern. The leaves come out to the sides and the leaf bases sort of clasp that main stem. But these are really cool, cute little things to see either in a stream or along a stream. And this is what those Fissidines grandifrons plants look like if they're dried, as in this herbarium specimen. So that's Fissidines. And I will finish up with Phallus liverworts, such as Connocephalum and Rabulia, and there are other genera as well. So these aren't necessarily going to be submerged in streams, but quite often growing on the margins or on stream banks. And you can also see a number of them if you were boating or paddling along sandstone bluffs at Mirror Lake State Park or at along the Wisconsin River too. So the Phallus liverworts have sort of this ribbon-like body. There are also leafy liverworts that look a lot like mosses that I'm not going to cover here, but there are different kinds of liverworts. So these Phallus liverworts have sort of this ribbon-like body, and they have specialized reproductive structures. You see some of them here and here. Sometimes they're up on little stalks and look like mushrooms. So these are quite interesting to look at. And so this is a liverwort called Connocephalum Conicum. It has a couple of common names, either the great scented liverwort, because it has a kind of a nice odor. If you pick a little piece up and crush it, it has a nice scent. You can also see why it's called the snakeskin liverwort. It does look like has almost like a snakeskin pattern on that phallus. But these are other cool little things that I think are a lot of fun to see if I'm out at a river or a stream or a lake. And I hope that that gave you an introduction to a lot of little fun green things, and you will be able to see them the next time you're out either monitoring your sites or just enjoying our aquatic resources. So I think we've got plenty of time for questions. If you have any questions, feel free to contact me. This is my email address, or if you have questions about blue-green algae or just want to explore more, this is the DNR's blue-green algae website. Thank you.
Well, Gina, thank you so much. We have a bunch of questions in the chat window.
All right.
The first one, I had a message sent just to me that I'm going to bring to you. This person was wondering if you had any recommendations for a magnifier that works with a smartphone to capture images of algae.
I don't have any experience with that. So I think just poking around in the internet to see examples of what other people have put together is going to be your best bet on that.
Okay. The next question is referring to species of algae that are toxic to dogs. Can you touch on that, please?
That would be cyanobacteria, otherwise known as blue-green algae. So not all blue-green algae make toxins, and not all of them do all of the time. But unfortunately, a lot of the ones that commonly form blooms in lakes and rivers in Wisconsin can produce toxins. So my rule of thumb, I have a couple of dogs who are behaving. They're not barking today, fortunately. But my rule of thumb is if the water looks like something I wouldn't want in my mouth, my dogs aren't going in the water, because if you're a dog owner, you know that they will drink anything, they will eat anything, they will wade into anything. And I guess the good thing about planktonic cyanobacteria is that if there is a whole lot of it, and you could potentially have high toxin levels being produced, if you have a lot of it, you're going to be able to see that. So if you've got floating green scums, mats, a lot of particles in the water, that's something that you can see. And that's when you really want to keep the dogs out. Now, if the dogs do go in, do your best to make sure they don't that they don't drink the water, and get them out as soon as they can. And it's always a good idea to wash them off after they've been swimming, just in case there is anything on their fur, so they don't lick that off and consume it that way. But the Department of Health Services has some excellent dog fact sheets regarding blue-green algae or cyanobacteria. So look at the DHS website for those resources under the resource tab.
Okay. Are there any green algae that we know that are harmful in any way?
Not as far as toxin production goes. You could argue that some of the real coarse species of Clydophora, the coarse species of Clydophora glomerata in Lake Michigan, you could argue that those are harmful and that they form nuisance growths. There are other green algae such as Pithophyra. Its common name is blanket weed. They can grow to really high levels, and anytime you have really high levels of growth of any sort of algae, if it breaks down, you can have reduced oxygen levels in the water as decomposition takes place. You get overgrowth of algae. It alters habitat. If they're not really a good food source, that kind of alters resources available to other organisms via the food web as well.
Okay. Next question was about a peak week for blue-green algae blooms in southwest Wisconsin. Is there a particular week that tends to be the worst from your experience?
I think someone is going to make a vacation reservation. I can't tell you for sure, because it's going to be different every year, and it's going to be different on every individual water body. And even within a water body, it's going to be highly dependent on a lot of weather conditions. So if you've got really calm, warm weather that's following heavy rains, you have great conditions for growing blue-green algae blooms. The rains have brought nutrients into that water body, and warm weather gives them warmer water, which they really like and do well in. And then that calm weather, if you don't have a lot of waves mixing them into the water, they're going to be accumulated at the surface as a floating scum. Wind is going to be important as well. If you have wind that's out of one direction for several days, it can basically transport the cyanobacteria across the lake to the downwind shore. And you may have really awful scum to the downwind shore, but upwind, it might be great swimming conditions. So when we typically see the most blooms, I think when I've been the most busy is really, I'd say mid-July into mid-August, but it can vary by individual years. So I can't give you an exact week.
Okay. The next question refers to the relationship between algae and fish. So can you touch on how algae relate to fish populations, how they support fish populations, or indirectly support fish populations, maybe through invertebrates or other food web relationships?
Sure. Well, algae are the base of the food web. So they're primary producers. They do photosynthesis. They fix carbon dioxide into carbohydrates and other compounds that then feed microscopic organisms, which then feed small fish, which feed medium-sized fish, which feed large fish. So it all gradually transfers up that chain. I will say diatoms are a really important high quality food, and they actually make an oil as their storage product from photosynthesis. And that oil basically accumulates up the food web, and that's what makes fish smell fishy. If you ever find a slimy rock that has a lot of diatoms on it, you rub your finger on it, you will end up smelling something that smells really fishy, and that's because of that oil that the diatoms make.
Cool. All right, the next question is regarding a possible link between cyanobacteria exposure and clusters of amyotrophic lateral sclerosis in humans. Do you know of that relationship, or can you comment on that?
Yeah, there is a large research group that has been working on a hypothesis that there is a compound produced by cyanobacteria called, it's a long name, the acronym is BMAA. A link between BMAA, which is a non-protein amino acid, and ALS, as well as other neurodegenerative diseases. I will say that this is not it's not fixed science yet. There are still a lot of questions around it. How one compound can possibly be involved with a number of different disease onsets. And there have been some opposing reviews in the scientific literature that have basically looked at what this research group has done and said, yeah, we don't agree with your analysis, and we don't think that you have showed a link between this. So I'd say for now, the verdict's really not settled yet. So it's still a very active area of research. And I will say that the research group is coming up with some very different and interesting prospective treatments for a lot of these neurodegenerative diseases. So I guess stay tuned.
Okay. I love the next question because I think it's going to be a fun answer. Why is Phanathystagnina one of your favorites?
It's macroscopic. You can get a lot of them floating up from the bottom, and people freak out, and there's lovely little grape-sized blobs of cyanobacteria fun, I guess. Yeah, so that's, I mean, if you wonder why I have the title for the talk, then that's kind of goes to show that I think these weird little green blobby things are a lot of fun. It's one of my favorites. I will say nostoc is my absolute favorite, though.
I remember finding that colony of Phanathysi that you showed on your slide, and I thought it was pretty cool because I was doing a plant survey and I expected it to hit a bunch of gravel because I could see down there and it just looked like a bunch of small stones, but it turned out the whole bottom was a Phanathysi colonies rather than rocks.
Yep. Yep. Yeah, there's a similar species of nostoc that grows to like be grape-sized. The common name in Danish translates to lake plums, and I've seen photographs of Arctic lakes that have it, the bottom's just covered with these. I think that would be my ultimate vacation goal would be to go to one of these lakes and see this for myself. Unfortunately, it's really hard to get up to the Arctic to get to some of these lakes.
Well, we have several more questions remaining, so let's try to get through a few more. How are the populations of different types of algae changing over time in Wisconsin lakes and rivers? Regarding climate change, maybe land use, other factors like that.
That's a good, huge question that I don't know if I can quite do justice to it here. Well, I don't have a great blanket answer. I think there are still a lot of high quality lakes with excellent water quality that really don't have a lot of change in their algal communities. Where we definitely see sort of this shift from diatoms and planktonic green algae to cyanobacteria tends to be in lakes where we have more nutrient pollution. So phosphorus and nitrogen has been coming into these aquatic systems from various sources. So either point sources or runoff from both urban and unused lands and agricultural lands. And that really is, I think, where we've seen the greatest shift. And where climate change comes into play is that the changes that we're seeing in Wisconsin, so things like more intense rain events more frequently and longer growing seasons, unfortunately, are starting to foster the conditions that can help cyanobacteria blooms to persist in our lakes.
Okay. Emily's wondering what the liverwort you mentioned, Connocephalum, what does that smell like?
Oh, I don't know if I could really describe it. It's a little bit floral, but it's just sort of its own smell. You'd have to smell it for yourself.
It smells like a liverwort.
Well, it smells better than you think a liverwort would smell.
All right, let's see. We have 1 minute left, so I'm gonna grab, we've got two questions about brightly colored algae. One of them, hot pink algae collecting by the springs in the bottom of a lake, and bright orange algae growing near stormwater outfalls. Do you know what these pink or orange ones might be?
I think I know. So I think both of those are not algae. So for the hot pink stuff near springs, there are organisms called purple sulfur bacteria. They look like they might be algae, but they're actually a type of bacteria that you typically see around springs and lakes because there's more of the sulfur compounds coming out with the groundwater there that they use as their fuel. So I would say hot pink stuff's purple sulfur bacteria. For the bright orange stuff, that is another kind of bacteria, probably iron bacteria. They typically can sort of help iron to precipitate out of groundwater, which kind of gives it that rusty orange color. So that's my best guess without seeing photographs, but just from my experience of people sending me pictures of things that look like those colors and those sort of locations, that's what I think they are.
Okay, wonderful. Thank you, Gina. We have other questions in the chat. If you don't mind, Gina, I will send all those questions over to you in an email, and you can respond to those. Thanks again to Gina for presenting, and thanks to all of you for joining us today. Remember, we do have one more webinar remaining in this series. It's called Watershed-Based Fisheries Management in the Driftless Region with Kirk Olson, a DNR fisheries biologist out of La Crosse. So join us for that webinar at 6 p.m. on February 17th. With that, I hope everyone has a great evening, and we hope to see you back here on the 17th.